Welcome to TWiM. It's awesome. That may be the best one yet. Hi, everybody. I'm Vincent and this is TWiM, This Week in Microbiology. This is episode 164, and today is November 3rd, 2017. You're listening to the podcast that explores unseen life on Earth. Today, I'm coming to you from Bloomington, Indiana. My first visit here. Never been here before. I'm at Indiana University, and I'm here to do this TWiM, and I'm also recording a TWiV later on, and Kathy Spindler's here for that. And in between, I'm going to talk about communicating science. And we have a great audience here today, as you heard. And if you have a chance for my seminars at 2:30, I think, right? You should come because it's not a data seminar. It's something different. I want to talk about what we do, communication, why you need to do it. And so if you have time, please come. And I think you'll enjoy it. Anyway, I have a couple of guests that I've selected from the faculty here. Since this is This Week in Microbiology, I've selected some people who work on bacteria. Who thinks that viruses are microbes, by the way? Kathy Spindler. Yeah, I think they're microbes. I'm in a microbiology department anyway, so they must be microbes. But for TWiM, we have three relatively new faculty members here. On my left, Shindong Wang. Welcome to TWiM.
Hi, everybody.
And next to Xindan, Ankur Dalia.
Hello, thanks.
Welcome to the podcast.
Thanks for having me.
And all the way at the end, Julia van Kessel. Thanks for coming, Julia.
Hi, everybody.
Welcome to the podcast. Do any of you know these individuals? Is anyone in any of their labs? That's good. Only a few, and the rest just came. So this is really great. We have a great audience. I'm really happy to see you. Now, we have an hour, which, as you know, if you listen to any of our podcasts. Who listens to any of our podcasts, by the way? A good number, almost half. So the others who don't, you should try listening. And as I always say, just subscribe anyway, even if you don't want to listen, because having downloads really helps us to do what we want to do. So I'm going to talk with these three faculty members today, a little bit about their histories and a little bit about their science. And we're going to see if we can do that in an hour, because that's when we have to get out of here. So first I want to hear about like where you're from and your training and so forth. So let's start with you, Xindan. Where are you from originally?
So I was born and raised in China. I did my first degree in Shanghai, where I studied human genetics. I was studying the association of some genes with schizophrenia. So back then, I was doing a lot of PCR and do sequencing and see if certain genes are associated with the disease.
This is in high school?
Yeah, this was my first degree, my bachelor's degree. And then I found that this research was exciting, but it was just too complex. If I want to tweak the system and make the mutations and study whether this gene is actually contributing to the disease, is too hard in humans. So I decided to go to the extreme and study bacteria. So then I did my PhD at Oxford in England and studying chromosome dynamics in E. coli. And from there, after graduating, I decided to stay with microbiology and stay with bacteria, but do similar research, but in a different organism, so Bacillus subtilis. So that's where when I did my postdoc at Harvard Medical School with David Rudner, and there I was doing research on chromosome biology, and I was there for almost 7 years. And then I came here at IU Bloomington in this February, and here I am.
February this year.
Yeah, this year.
Wow, I think this is the youngest person we've ever had on TWiM, or the youngest faculty member. That's great. So what got you interested in science way back when? Do you know?
So I think I was sort of born as I wanted to do science when I was very little. So just right when I learned how to do additions and subtractions, I just got into numbers. So I was very fascinated by the math and how to do this you add something together, it gives you something, and then you subtract. So I was very interested in numbers just when I was very little. And then I discovered the logic of mathematics. And later, I found that mathematics is not only useful just to do number work, it's also useful to do physics, and then chemistry, and then later biology. So I just felt I was just very intrigued with solving problems. So I could sit there for hours and work on a problem. And then when I finished it, I want to find different ways of solving it and find a better way to do that. And I got inspired by my teacher at middle school, and he gave me a book with all these interesting problems to solve. And then I got a book from my parents' friends about 100,000 whys. So just
100,000
whys. Yeah, it's like an encyclopedia, but with very simple questions. So I just got into science very early on, and I think how I get to where I am now is kind of to the specific research I do is kind of by chance. At certain points, I met certain people and I got inspired and went to this particular path. But if I didn't do this science, I would end up doing something else, some other science. I'm driven by my curiosity, and I will always try to find out why. And
it's funny, 100,000 whys. We always say on TWiM, you can't ask why questions. You don't know why things happen.
Right.
So we should sequence your genome and see what genes are associated with being born curious about science. That would be great. So why did you come to Indiana University?
Well, there are several parts of this story. So the first one is the year when I was on the job market, IU was hiring. So I applied for the job and they gave me the offer. So this is part of the story. If this didn't happen, I wouldn't be here. So the second part of the story is I also applied only to a handful of positions. I only applied to the positions I really wanted to go. And the last part of the story is, in the end, I also have to choose from this offer and a separate offer. So I have to decide which place I want to go.
Yeah.
And so that other school actually has a better ranking. Maybe this means a lot for undergraduate students. And it's also at a more attractive location. And it has a nice department. It has all the equipment I needed, facilities. But in the end, I decided to come here. And the reason is the things we have here, they don't have, is the microbiologist community. So we have a large group of microbiologists doing exciting stuff. And so I feel my expertise could be more useful, be explored more by the faculty around here. And people doing the work they do, I could also use in my research. So I feel I'm a better fit in this position. So here I am.
So was it the right decision?
Yeah, never regret.
Great. Ankur, where are you from?
I'm originally from New Jersey, and so
Where?
I'm from Bergenfield up north.
Oh, that was
It's right next to Paramus. And
yeah, I know it well. I was born in New Jersey, and
oh wow,
I
okay.
grew up in Bergen County,
Oh wow,
so I know Bergenfield.
yeah. So all right, Jersey boys. All right,
Yeah,
great.
we're Jersey. That's right. That's right.
Yeah,
I have a stronger accent than you.
Yeah, I guess so. That's the one thing I'm told by all my students, that I talk too fast. I blame that on Jersey all the time. So yeah, so I grew up in Jersey, and I went to undergrad at Cook College, which is part of Rutgers University, and that's where I really started kind of doing science. Worked with Don Kobayashi there, working on Enterobacter cloacae and selenate reduction. And from that experience, basically the one thing I knew was I liked bacterial genetics. So I decided to kind of pursue that further. So I went to grad school at UPenn, working for Jeff Weiser on Streptococcus pneumoniae, looking at the host-pathogen interaction. And while that was fun and kind of interesting, I didn't really like the host side of things. I was really more interested in the bacterial side. And so I decided I wanted to kind of further push bacterial genetics and see if I can pursue that further. And so I tried to find a lab that was kind of doing cutting-edge bacterial genetics. And I found Andy Camilli, and who was looking for a postdoc, so it worked out perfectly. And so I did my postdoc with him for about 5 years until made the switch to kind of go on the job market, and luckily ended up here.
And when did you come here, roughly?
2 years ago.
2 years?
Yeah.
So we had Andy Camilli on a TWiM some time ago.
Oh, okay.
And I was just at Tufts 2 weeks ago recording a TWiM also.
Okay.
So there's all sorts of coincidences. So for you, what catalyzed your interest in science? How far back can you figure?
So I mean, since I was a child, I guess I was always interested in taking stuff apart and putting it back together. I wasn't very good at the putting back together part, I guess. But I slowly got better at that. And then I think what really kind of gelled the idea of academia as kind of a science position, or kind of going into this field, is when I was an undergrad working with Don Kobayashi. I remember thinking at one point, oh my God, he gets paid to do this? And I was sold from that point on. So I kind of always wanted to end up doing what he was doing, and luckily I've been able to, but never know if you can at the time.
He gets paid, but not very much, as we all do. So why did you come to Indiana?
It's the environment. I'm going to mirror, I guess, what Xindan said a little bit. Yeah. But basically, I had a couple of offers to look at, but in terms of the environment and the people here, the science that's being done here is truly cutting edge, and the collisions and kind of collegiality in the department is unparalleled in my opinion. So I've already had opportunities to interact with a number of faculty. We've published papers together, and in the short time that I've been here. So it's paid off in spades.
And you're glad you came here?
Oh, absolutely.
I can't ask him. I really shouldn't ask him that question. He's not going to say no. But you can. Yeah, that's right. We'll wait till after tenure and say, is it the right place to be? Okay. Julia, what's your story? Where are you from?
Right, so I'm from New York. I went to undergraduate at
New York, what part?
It's near Poughkeepsie. It's a tiny little town called Millbrook.
I know Poughkeepsie well. My son goes to Marist.
Oh, yeah, absolutely.
You know where that is?
And Vassar's there as well. And it's
it's a nice part of New York.
It is.
So you grew up around there?
I did, yeah.
Neat.
And then I went to undergraduate at a small school called Utica College, which is in central New York. And I was really lucky. There was one microbiology lab in my very small college, and the person that ran it is Lawrence Aronson, and he was incredibly generous with his time. He's one of the best mentors I've ever known. And I was really lucky to study with him, and we looked at the microflora on red-backed salamanders. It was really fun. So I really got into bacteriology that way. And I went and did my graduate work at the University of Pittsburgh in the lab of Graham Hatfull, where I studied Mycobacterium tuberculosis and bacteriophages. And then I decided I did not want to work in a BSL-3 facility for the rest of my career. So I was looking for a change, and I went to an American Society for Microbiology conference, the general meeting, and I saw Bonnie Bassler talk, and I was instantly starstruck, which if you've ever seen Bonnie talk, you would understand. So I wanted to study quorum sensing, and I was lucky enough to get a position in her lab as a postdoc. So I was at Princeton for about 5 years, and then I came here. So
when did you come here? How long?
About 4 years ago, actually, now. So. And
why did you come to Indiana?
So we looked in a lot of places, I think like many people do, much like Xindan and Ankur. So we had what we would call a two-body problem. So my husband's a biochemist in the biochemistry department here. And it's not easy to find two positions at the same time, but we did, and we are very thrilled and lucky to be here. Again, same reasons as Xindan and Ankur. the community is fantastic. And just the kinds of science that we can do here, we've just learned so much in just a short amount of time. The new kinds of experiments and methodologies and collaborations, I don't think it's not possible anywhere else.
So I always love to point out the connections. So I know Graham very well.
Yeah.
We had him. So here's a story, quick story. I was giving a talk in Edinburgh many years ago about communicating science, and he sat in the front row. And he raised his hand. He said, "Do you ever talk about phages?" And I knew who he was. I said, "Why don't you come on and do it?" So he did. And we talked about his C-phages program and so forth. So that was good. And then, of course, Princeton, as I told you, when we work on our virology book, we go to Princeton to do it. And I was just there last week. We're starting again to do it. So it's a great place.
It is.
I love Princeton. So that's great. All right. So there you go. There is, and for you, it's probably been the right decision also to come here.
Absolutely.
Right? Let's talk a little bit of science and find out what you do. Shindong, let's start with you. I know you have this wonderful website. If you haven't seen it, you should go. She's got a .com of her own. What is it? What's the URL? Tell us.
Oh, just my name, wangxindan.com.
Wangxindan.com. And so it takes you away from the university, but it's really well done. And I told her it's mobile friendly, which is really important these days if you want to make a website. And what did you tell me?
Thank you. But this is thanks to my husband, who is a web designer and developer.
Yeah. So if you want a website made, you should check out his work. And I might use them too, because I can use some web design as well. It's really well done. Why did you do that? Why did you make your own little site off by itself? I'm wondering.
So I've had it since 2006. So back then, I just wanted something unique, something that is not like any other website. Yeah. And I asked my husband to make one for me, and I've had it all the time. So it has changed a lot. So every couple of years, I change it completely. Right. This is a new phase, but I just feel it's like my personal little world. I can put anything there.
I think this well, almost anything, right? I think it's a great idea. For those of you who are just starting out or early on, it's a great idea to have a website where you can put things, you can write or talk about your experiences in science, and then people can find it. And I think that's one of the ways you can communicate what you're doing. So as you said before, you're interested in chromosomes and how they move around in bacteria. Why don't you tell us big picture of what you're interested in?
Yeah, my lab studies how the chromosome is organized inside living cells and how this organization is passed on through generations. So we study a set of protein factors that are important for this organization, and we study how these factors structure the chromosome. So some of these proteins are conserved from bacteria to humans, and they are found in many organisms. And we hope that by studying these proteins, using bacteria as a simple model organism, we could uncover some fundamental mechanisms of chromosome folding in all organisms. And this is sort of the general theme of my labs.
So you use Bacillus subtilis as a model, right?
Right.
And why not E. coli or some other bacterium?
Yeah, so that's a good question. So actually, I did my PhD studying chromosome organization in E. coli. And in the end, I decided to switch. So for several reasons. The first one is, in most bacteria, chromosome segregation is driven by a conserved factor called the partitioning system. And this factor is present in about 70% of the genomes. And E. coli is the outlier here. It doesn't have the part system. You may say that if there are certain percent of bacteria that don't have them, maybe they are not important, so why bother? But there are also the 70% that have them. So they make this segregation process more efficient, and I want to study how they does it. So how they do it. So this is first the reason, the partitioning system. The second reason is bacillus, when they grow out of this exponential phase, when they are sort of in nutrient limiting state, they differentiate into different cell types. And the one type is sporulating cells. They form a spore so that they can resist many environmental harshness.
By the way, are spores alive?
Spores, it's sort of dormant, but they are also constantly sensing the environment. And when there is some triggers, when there is good food around, they will germinate like a seed and try to make more of the cells. So they have sporulation cells. And they also, some cells decide to take on genomic DNA and become competent and try to make take new genes in their system to be better.
That's
right?
what
This is
Ankur works on, right?
So this is a competent state, and they can also so if they sense some surface, they can also move in a certain way. So the swarming motility. So bacillus just have all these different cell types, and by microscope you'll see that the chromosome can adopt unique or some special conformation that you don't see in other cell types. So I like to study bacillus because I want to look into the chromosome and see how they are folded in these different states and what are the factors that make them fold like this. And so this is the second reason I got into bacillus. The third one, I only knew this when I am already in this field, is working with bacillus. It's so easy. It's so easy to make genetic manipulations. And
easier than coli?
Yeah, a lot easier because of the natural competence. You grow the cells, you take the DNA, and you make the
So you don't have to make them competent as you do E. coli.
You just grow them to a certain 4 hours in a media, and you add the DNA and they take it. So it's just so easy. So
what is now listen to the way I phrase this. What is the reason that they are, and Kathy's laughing because she knows why I'm doing that. What is the reason that they are competent all the time and not sometimes like other bacteria? Do you know?
So they are not competent all the time. Okay. So when they grow exponentially, they are not. Only when they are sort of stressed, there are some signaling pathway turned on, and then they thought maybe need to change. And yeah.
Okay, so tell us what happens to the chromosome during a growth cycle of bacillus. So when they're growing and then when they divide and all that, and how that relates to what you're interested in.
Right. So I need to step back a little. So most bacteria has a single chromosome, and it's a circular DNA molecule. It has a replication origin here, the terminus here, and the 2 replication arms like this.
It's circular though.
Yeah, it's a circle. It's a one circle. You have origin, terminus, and the 2 arms.
And for B subtilis, how long is it roughly?
Yeah, it's around 4 million base pairs. So the first study of a systematic study of a bacterial chromosome segregation was in a model organism called Colobacter crescentus. So this DNA molecule in a rod-shaped cell is compacted 1,000-fold. But we need to think about it. It's really like shoving a rope of like 10,000 kilometers or 1,000 kilometers in your backpack. It needs to be compacted a lot. But the cells are growing very fast, and then they have to constantly make new proteins and respond to all these different stresses. So it needs to be organized. So, and it does. When people look at the chromosome organization using this bacterium, they found that this chromosome has the replication origin located to one end of the cell and terminus to the other end. And the 2 replication arms are aligned linearly from one end to the other.
So there's one origin.
Yeah, so one origin to one end, terminus to the other end. The 2 arms are just
When you say 2 arms, you mean the replication is bidirectional from the origin?
Yeah, yeah. So And then this was just a very intuitive organization. You have the chromosome, you just organize like this. And this was thought to be true for all the bacteria. So when I started my PhD, some observations in E. coli didn't fit this regime at all. So I decided to look into the chromosome organization in E. coli. I found that in E. coli, this organization is rotated 90 degrees. So basically, the origin, instead of at the pole, is now in the middle of the cell, and the two replication arms are on opposite cell halves. And this is quite very different from what we thought before. And then during my postdoc, I moved on to bacillus. I found that in bacillus, the chromosome actually adopts an unusual dance. So at the beginning of the cell cycle, the two arms are like this, but some point in the cell cycle it's rotating like this. So every cell cycle, it does this oscillation. So then, so later and right now, I'm trying to find what is contributing to these different organizations. Actually, all these bacteria use very similar set of factors, and they use it slightly differently, and now they result in these seemingly diverse organization patterns. But basically, they have a very fundamental similarity between them. And then that is that they are compacted in a certain way, and they are localized linearly. And all the DNA replication, segregation, and cell division are happening at the same time, and they have to find a way to coordinate this. And
So is the chromosome attached at all to cell wall or anything like that?
So in some bacteria, it's very well studied that the origins are attached to the cell pole, and so it might be attached all the time to the membrane, but we don't know this for sure
bacillus,
yet.
you mean?
Yeah, for bacillus, there is attachment at certain point in the development, but it's not clear that when the attachment is happening all the time.
And I know in E. coli, at least, there's growing understanding that there's a cytoskeleton,
Right.
And that's involved in division and partitioning. Is this the case with bacillus as well?
It can be. So seemingly everything is connected, and we are just now trying to find the connections and trying to make this,
Right.
Yeah,
Okay.
trying to find this connection, and we discover it more and more. Yeah, but it's not.
All right, we'll come back to you. Let's move on to Ankur so everyone can talk. So you work on Vibrio.
I do, yeah.
Tell us just a little bit about Vibrio cholerae, right?
Yeah, yep.
It's a pathogen,
It is
right?
a pathogen, yeah. So it's the causative agent of the diarrheal disease cholera. And so it's a devastating disease. Normally it's only kind of affecting populations in the developing countries, really where people lack access to clean drinking water. And so if the bacteria is ingested by a human host, it'll colonize the small intestine. It'll elaborate a toxin called cholera toxin, which causes the disease, basically, is a main proponent of the disease.
So where would you acquire it from? You said contaminated water.
Correct.
Right?
Food or drinking water is a common route for infection.
So, but it lives in the environment also, is that right?
That's right, yeah. So it's normally environmental and lives in kind of brackish water environments is where it kind of likes it the best. And when it's in the environment, one of the things it likes to associate with is the chitinous shells of kind of crustaceans. So, and that compound of those shells is called chitin. So if you've ever had kind of lobsters or crabs, the shells of those organisms are made up of chitin. And so if Vibrio is kind of in the environment floating around, it'll find a piece of chitin, latch onto it, and form a biofilm, divide, and grow on that surface.
So you said they're mainly in brackish environments, right?
Correct.
So you wouldn't find them in the open ocean at all?
Vibrio cholera, I think, could also definitely be found in the open ocean. It can tolerate high salinity, but I think it thrives in brackish water environments.
So if you could show me a picture of the Earth and tell me where Vibrio cholera was, where would it most likely be?
So I guess I have to disclaimer this a little bit. So Vibrio cholera, there are two types of Vibrio cholera. There are Vibrio choleras that are completely environmental, will not cause any disease.
Okay.
And then there are toxigenic Vibrio cholera that have a specific element, which is a phage, basically, that's lysogenized in Vibrio that encodes the cholera toxin. And so those are called toxigenic Vibrio cholera strains.
And that's what you work on.
Correct. And so normal Vibrio cholera are very broadly distributed across the globe. Toxigenic Vibrio cholera are much more limited in kind of their distribution.
And Julie is nodding because she also works on a Vibrio, right? But it's a different Vibrio.
Yeah.
So make sure he says everything correctly. Because I can't catch it, that's for sure. So this phage that was acquired happened a long time ago, presumably.
Correct.
Right? And does it confer some advantage to Vibrio?
That's a great question, actually. It confers the ability to cause disease. Whether or not that's a selective advantage or not, I guess, is a question.
Right. But if you take the environmental Vibrio and put this phage in them, will it make them pathogens?
So they need to have the right receptor on the bacterium for them to take the phage in.
Yeah.
And so I don't know if that experiment has been done. Actually, I'm sure it has. I just don't know the paper that describes it. So I don't know the answer to that question, actually.
So the toxigenic Vibrios, they live mainly in brackish waters, is that right? Or
They can, yeah. So they'll thrive in brackish water environments, but they can also survive in basically straight water as well, but they'll also survive in higher salinity. So they kind of have a salt tolerance that allows them to live wherever.
All right, so one more general question.
Sure.
So when there's an outbreak of cholera, so and Haiti was brought there,
Sure.
right? But are there any instances where someone actually acquires infection, not from a human to human transmission, but from an environmental source?
Absolutely.
Yeah.
Yeah, that can certainly happen. And that actually happens cyclically every year in Bangladesh, is an area where cholera is endemic. And so it's thought that kind of Vibrio cholera is sticking around in the water, and every year a new outbreak kind of comes out. And if you look at the strains that are causing disease every year, they can differ from year to year, suggesting that they are acquired from the environment, yeah.
Now, from what I understand from your website, you're interested in how the Vibrio take up DNA.
That's right.
So
yeah.
tell us what you're interested there.
Sure, yeah. So I mean, this process that they use to take up DNA is called natural competence, as kind of Xindan alluded to. And so this is a way these bacteria, when they're in the environment, they will induce themselves for this process. They'll make this protein apparatus, basically, to take up DNA that's sitting outside of the cell, bring it into the cytoplasm or inside the cell, and then recombine that DNA into the chromosome. And they can use this as ways to, acquire novel traits or to introduce mutations to existing genes. And so my interest is really pretty broad in terms of this process. I really want to know how it works in a fine, detailed way. So we're really interested in what are the mechanisms these bacteria use to take up the DNA, what are the mechanisms that they're using to actually integrate in the chromosome, and even further back, we're really interested in looking at the regulation of this process. How are they controlling the processes that allow this to happen?
So this uptake of DNA, this implies that there's DNA everywhere, right? Is that true? If you go into the environmental niche of Vibrio, there's lots of DNA around? Or
So the way that these things, I'll back up and describe, I guess, when they're turning this system on. So they're normally turning the system on when they're forming biofilms on chitin, which is that thing that I mentioned that they're interacting with in the environment. And so I guess the idea there is that when they're colonizing chitin, it's very likely that you'll be surrounded by neighbors, potentially other Vibrio species or other Vibrio cholera isolates, and it's a great environment in which they can start to share DNA potentially.
So if they are in someone's intestine, are they forming a biofilm and turning on these DNA uptake?
Great question. I don't think anyone knows the answer to that yet. And so in terms of thinking about chitin as an inducer for this process, or normal, chitin biofilm formation is what normally turns the system on.
Yeah.
If there's a chitin biofilm in the gut, that could potentially turn on this system, I would say. But it's also known that if you give the bacteria other carbon sources, that'll prevent them from turning the system on. So if there are other preferred, pieces of food around, they'll actively not kind of turn the system on. So it's hard to say whether or not it would turn on the human gut or
But
not.
we don't have chitin in our gut, do we?
We do not, no.
So that it's not going to be a chitin-dependent process, but it could be dependent on something else, as you say.
That's possible, sure.
Right. And so if would Vibrio be on the lobster shell, for example, or not likely to be there?
It certainly could be. Yeah.
What other
if you're getting
sources
that
of-
lobster from the wrong water source, I guess.
No, but in the ocean,
possible.
would Vibrio? What other sources of chitin besides crustaceans, for example?
Oh, so I mean, it is mainly crustaceans, but it's not going to be the macroscopic, huge crustaceans like lobsters and crabs. They're also colonizing really microscopic crustaceans like zooplankton as well.
Okay, so the transfer of DNA, is that from other Vibrios or other species entirely different?
It could be from potentially other closely related species as well. So that the way that DNA is actually integrated in the chromosome is using homologous recombination, which actually means that DNA has to be somewhat similar to the host chromosome for it to actually be kind of crossed in and integrated. So it could be other Vibrio cholera, isolates that are out there in nature, or it could be other Vibrio species that have similar genetic content.
So if you look at the genome sequence, can you tell sort of the origin of different parts of the genome?
So actually, there's not a ton of examples of movement of this DNA kind of among Vibrio cholerae isolates in nature. And so it's hard for us to get a consensus on where pieces are coming from currently, basically.
Yeah. Oh, okay. So you're interested in how chitin regulates this, is that right?
Correct, yeah.
Tell us about that.
Yep. So I mean, so these bugs, when they're sitting on a piece of chitin, they basically have to degrade this compound. So chitin is an insoluble polymer of N-acetylglucosamine, which is just a sugar. So it's really just a chain of sugars that is normally insoluble. And so for Vibrio to kind of gain access to this awesome carbon source or this sugar to eat, they need to degrade that carbon source into soluble sugars, and then they eat that basically. And so in order to kind of regulate that process, they don't want to turn on the enzymes to break that sugar down all the time. They have a system in place where they can sense some of those sugars and then crank on the expression of these genes that are important for eating the chitin, but also turning on this process of natural competence. And so we're really interested in trying to figure out what are exactly the enzymes that mediate this process. How are they sensing those sugars to turn on these distinct processes of eating the chitin versus turning on competence, and is there interplay between those systems?
So the use of chitin as a source and the uptake of DNA are both linked.
Correct.
Right? Is that coincidental, or do you think
I think it comes back to the question of when should you turn this mechanism of DNA uptake and integration on? And I think that in terms of the ecology of the organism, it might make sense to do it under conditions when they might be surrounded by neighbors. And growing on a chitin biofilm is a great environment for them too. I mean, this is anthropomorphizing the bacteria, right? So I don't really know what Vibrio cholerae is trying to do, but it makes sense in the sense that it could be a good environment to share.
So how do you study this? Do you grow Vibrio on crustacea in the laboratory?
That's right, yeah. So basically, we actually order from Sigma, very, very, fancy compound, crushed shrimp shells that we use for our experiments in the lab setting. And actually, the batch of shrimp shells that we get have to meet certain criteria. We had a scare in the lab where we thought our shrimp shells were going off, so we had to find a new lot of shrimp shells. But yeah, so basically we grow them up in some nutrient broth and transfer them over to a tube containing just some crushed shrimp shells, and they turn on this competence machinery, yeah.
And so you study what genes are going on? And so
That's right, yeah. So what genes are going on, what are the genes that then turn those genes on, and what is the mechanism of everything in between, basically.
So if you met someone in an elevator who wasn't a scientist and they asked you, what's the relevance of this work to me, what would you tell them?
Well, I would say natural transformation is a great way for dissemination of pieces of DNA, which could be antibiotic resistance determinants, they could be virulence factors, they could be other things that some pathogens use to kind of increase their virulence. So in terms of human health and biology, certainly pathogens have this capacity. We probably should try and understand it, because if we understand it, we might be able to stop it. And so that's, I think, one aspect to this for sure.
So there's no crustacean DNA in the Vibrio genome, right, I guess?
I don't know that I've looked for that, actually, but I'm going to say tentatively no.
Someone once told us years ago, DNA is a real problem. It's everywhere. If you're doing very sensitive sequencing, you get contaminants all the time of all sorts of things. So I have this vision that it's all over, and wherever environment you look at, there's DNA. So eukaryotes take up DNA as well, right? We're just beginning to realize that. So I think that's a really interesting way to look at it. All right, we'll get back to you. Julia, you also work on a Vibrio, but a different one, right?
That's correct. Yeah, we work on a Vibrio harvii mostly in the lab, although we work on Vibrio cholerae occasionally, sometimes some of the other marine Vibrios as well.
Do you guys talk to each other?
All the time.
Absolutely, yeah.
Are you near each other physically?
No.
Only one floor away, yeah.
Okay.
And there's a third Vibrio lab here too, which is Dean Romagnus's lab. So we have a great Vibrio community at IU.
Which Vibrio?
We have a quorum.
Yeah. We have a quorum. That's a good title, right? We got to somehow put Indiana in the title. What Vibrio is that third lab?
They usually work on vulnificus, Vibrio vulnificus, sometimes Vibrio parahemolyticus as well.
So harvii is not a pathogen, right?
It's not a pathogen to humans. It's a pathogen of shrimp and fish, and so more of a concern for aquaculture. But that's right. We get to work on a non-pathogenic one for the most part. We work on them, however, because they are bioluminescent, and that is one of the more important traits that we study in the lab.
So what's the function of bioluminescence in their life?
Yeah, that's a great question that nobody knows the answer to.
Really?
Yeah.
Huh.
So most of the ones that you hear about are from Vibrio fischeri, which we have we've as a community studied Vibrio fischeri bioluminescence and its role in symbiosis with the bobtail squid.
Right.
Vibrio harveyi, it is not clear why they bioluminesce. There are a lot of hypotheses. I don't think anyone has ever determined the actual reason.
But for fischeri, the luminescence helps the squid.
It does.
Right?
Yeah.
Do we know what it does for fischeri at all?
The bioluminescence? So the bioluminescence in fischeri is important for the squid to, it's a counter-illumination strategy.
Right, yeah.
Right, so they're in the ocean in Hawaii and picture this nice moonlight in Hawaii on the beach and they're in very shallow water and the moonlight makes a shadow and the squid needs to hide from its prey. Or predators and to also hide from its prey. And it makes a shadow, and so the light from the bacteria that it has in its light organ hides the shadow.
And so the bacteria get a place to live
And food,
and
most
exchange
importantly.
light for the squid, and that's the symbiosis there. Right?
Absolutely.
But we don't know for harveyi what must do something though. Right?
There are some ideas. One of them is that it is the bioluminescent bacteria when they infect, let's say, shrimp, and then the shrimp might then be bioluminescent, which they are, might be another way to then the shrimp are then prey for other predators, and that might disseminate the bacteria more. Some people have hypothesized that it's just an accident of its enzymatic light reaction. It just produces light. It's that's part of other metabolic processes. Could be that there's a symbiosis with an organism that we just haven't studied very well.
But you and others in the field use it to study quorum sensing. So can you explain quorum sensing?
Sure. So quorum sensing is our technical term for how bacteria communicate. It's how bacteria, they secrete molecules. These are called autoinducers. These are usually specific to the bacteria. They can also be more general to that many bacteria can detect. And it's their way of counting their numbers. So the number of these autoinducers in their environment is a proxy for how many cells are in the environment. More molecules means more cells, and the cells can bind these autoinducers and detect them and change their gene expression. So they change their behavior. And if they're in different environments, under different situations or around other bacteria, they can modify their behaviors accordingly. So we often think about bacteria as individuals, so perhaps swimming around in the ocean alone, right? Or like Ankur was talking about, living on a chitinous shell in a biofilm, and how their behaviors change under those circumstances.
The idea that bacteria live in communities is not very old, is it?
No, and it's really actually one of the first things I talk about when I introduce the idea. We always think about bacteria as these single-celled organisms, or at least that's how I was taught way back when. But we now know they exist in these really complex multicellular communities, and they're interacting, and they're sometimes fighting each other, and sometimes they're beneficial to each other, and it just depends on the environment.
There's now some evidence that viruses may induce cells to secrete substances that cause quorums to occur as well.
Yes, it's a very interesting new development in the field. There have been a couple very recent papers that have been really fun to read about that topic. Yes.
But you use luminescence as kind of a readout for quorum formation, right?
It is one of the best readouts.
Right, but there are other things that happen as well in response to these molecules, right?
Right, so that's actually one of the reasons we use Vibrios as models, because they do fascinating things responding to quorum sensing. So actually competence is one of them, like Ankur was just talking about. Biofilm formation, multiple kinds of secretion systems, motility, there are a number of things that change.
And are there many molecules that are produced that are used to sense quorums?
Yeah, so in every bacterium it's different. The structure can be different, anything from things with lactone rings to long carbon chains to peptides, depending on the organism. In Vibrios, there are, every Vibrio has multiple autoinducers, multiple molecules, multiple proteins that detect those molecules. It can be quite complicated in certain species.
Do most people who study biofilms use these model systems like harveyi?
So the best biofilm model is Vibrio cholerae, actually. So it forms very nice biofilms, and it's something that's highly studied. There are people doing some really fascinating work looking at how bacteria change gene expression in different parts of the biofilm and how those cells align in the biofilm and things like that.
Okay, so tell us what aspect of quorum sensing you're working on?
Sure. So actually, after I tell you everything about those molecules, we actually don't work with them at all. So we're actually interested in what happens downstream after all the communication has occurred. So we're interested in the proteins, the transcription factors specifically that enact all those changes in gene expression.
So when how do these molecules do that? Do they bind receptors?
Right. So in Vibrios, it's somewhat different than in most gram-negative bacteria. In most gram-negative bacteria, you have a molecule that is diffusing through the membrane and is bound inside the cytoplasm by a transcription factor. In Vibrios, we have those, but there are others, and some of the molecules are detected on the surface and then have a signal transduction cascade that then changes gene expression of a very important transcription factor at the center of the pathway. So that's where we've been focusing our research, is on the center transcription factor, and how this really one remarkable protein changes hundreds of genes, turns them on, turns them off. There's a massive cascade that happens. We're very interested in when those genes are turned on and off, and also how that ties into other signaling systems. So changes in nutrients or salts or different growth conditions, surfaces or not surfaces, and how that intersects with quorum sensing.
So one of these genes under the control of this protein must be the luminescent genes,
Absolutely.
right?
The bioluminescence genes is one of the first ones that was discovered. So we know a lot about the bioluminescence genes. We know a lot about their enzymatic activity. We actually still don't know a lot about how they're regulated. And so one part of my lab focuses very closely on the mechanism of gene regulation in quorum sensing in Vibrios. And the bioluminescence promoter is a great example that we work with. So.
And what does the other half do?
The other half thinks more about the downstream phenotypes. So I have people that are interested in how, after quorum sensing signaling, what changes happen. So let's say osmotic stress response genes, for example. These are bacteria that are marine organisms. They experience different changes in salinity all the time, and they turn on the osmotic stress genes when they're doing quorum sensing. We don't know why, but we're very interested in how the salt signaling and the quorum signaling intersect at these genes. There are other people that are interested in motility and biofilms. Type 3 secretion is another one that we study. So really looking at how multiple pathways intersect.
So do you think you could study this the rest of your career, or do you think you could be distracted if you found a particular downstream operon that was really interested and had nothing to do with quorum sensing?
Well, I do get distracted a lot, actually, but so that, yeah, I think the mechanism work is probably always going to be at the heart. The regulation of mechanism will always be at the heart of our work. It's really been driven a lot by what the individuals in my lab are interested in. So they kind of have their own favorite pathway and what physiological question they want to ask. So it might be that they'll get fascinated with one thing and we'll go with that for a while.
So you're okay with that if someone came and said, oh, this is really cool, I want to work on this? Yeah?
Absolutely.
Good. Now let me ask you the same question I asked Ankur. If you're in an elevator and someone asks you what's the importance of glowing bacteria, what would you say?
Right, so I always say this metaphor, if bacteria are alone in their environment, let's say they're bioluminescent, it's not going to do very much good. But if there's a trillion of them in one area and they turn on bioluminescence, what does that do? Well, for Vibrio harvae, we might not know, but we know a lot about for pathogenesis, right? So bacteria that are alone tend not to cause disease, but we know when there's a million of them in the human host that they cause disease. And so our research has relevance for studying pathogenesis in humans as well as other animals and plants. And what we're hoping to learn is how to turn off quorum sensing so that they cannot use their quorum sensing strategy to act and secrete virulence factors and make good biofilms. So if we can turn that off, maybe we can turn off pathogenesis. And what's really important about that is that molecules, and these are being developed now, the molecules that block quorum sensing don't have a very strong selective advantage for the bacteria to generate resistance. Right? So this has been pretty carefully studied that antibiotics have a very strong selective advantage to generate resistance. Molecules that target quorum sensing would ideally not do that. So there might be a lower rate of resistance.
Do we understand why that is? Why there's very little selection for resistance?
So generally, even if you knock out these quorum sensing proteins, the strains don't have that much of an effect. There is a slight growth disadvantage, but it's not nearly as bad, obviously, as a bactericidal antibiotic would be.
Right.
So.
Right. Okay. I didn't ask you, Xindan, that question. So let me ask you now. If you were in an elevator and someone overheard you talking about studying chromosome movement, what would you tell them if they said, why does this matter?
Well, in every cell faces this problem if they want to make two daughters. They have to copy the DNA and distribute them evenly to the daughter cells. And in bacteria, they do it very fast. Every 20 minutes, they have to do this. So it's very important for the bacteria to preserve the genetic material. And so from bacteria, one cell is the whole organism. And for us, every cell has our genetic material, and it's doing exactly the same thing. We face the same challenge. So chromosome organization, segregation are important.
Now, of course, you don't have to explain any of this to me or us in this room, but I think it's really important to let the general non-science public understand why we're doing what we do. Because we know that you could work on the most obscure thing and it would be cool and it might lead to something great one day, and we get that. But I think it's really good to put your work in terms of, an elevator talk. I think it's essential. So I want to ask you all some fun questions maybe. And Julia, you're all pretty early on in your careers, so some of these are less compelling, I think, than we'll ask later on TWiM. On TWiM, we have mostly old folk, I think, right?
Adam's offended.
You're mostly old, yeah. I'm old too.
Very young.
Mentally, sure. So, Julia, if you hadn't been a scientist, what else would you have done? Do you ever think about that?
Actually, I just like Xindan, I love math. I've always loved math.
Really?
Yeah, I actually wish I had taken more of it in college because I've apparently forgotten it all. But yeah, I really loved math.
What would you do with math, though?
Oh, I really like balancing checkbooks, probably accounting.
Oh, cool, accounting, great. How about you, Ankur?
I think I would have either been, so when I was going to undergrad, I was either going to go for science or going to medical school, or I was going to become an architect is my other idea. Because my dad's an architect, saw what he did, really enjoyed it. And so either architect or an engineer, I think, would be the only other career I would have considered. Something that I could still tinker with every day.
Right.
Yeah.
So you like to take things apart.
That's right. Yeah, that's the other half of my lab is we do a lot of, we develop methods for genetic engineering. So I'm not actually an engineer, but I can at least mess with the bacteria.
So when I was a kid, I also liked taking apart TVs. And I would take them apart, never put them back together.
We had very similar youths.
But it's New Jersey.
That's right.
We had, in those days, TVs were huge boxes with big tubes. And I would cut out all the components and then put them in drawers. And my mother would say, "What are you doing?" And at the end, we threw it all out. But the best part, we would take the picture tubes and put them in the woods and throw rocks at them.
Oh, and they'll explode.
And they would explode. How about you? I think you kind of mentioned you always wanted to be a scientist and nothing else. But is there anything else?
So, yeah. Let's say maybe a more realistic question you would ask is, what happens if you burn all your startup money, you don't get your funding, and you have to close your lab, and you can't do any science-related things, what would you do? So this is more realistic, and I actually think about it. So I think I would want to become a math teacher, and I would like to teach middle school students when they are still quite young and fresh and they don't know too much about life. And I like to plant this seed in their head early on, and I like to inspire them to find their talent and to be aware of their talent and to find what they are curious about and pursue it. I hope to let them know that there is something bigger than pursue something bigger than yourself and try to do a degree in STEM. So to get curious and try to ask questions, and even if I'll try to get them to do a PhD, I'll get into science. I want to let them know that even if what we are doing is not immediately related to human health, I'll try to make it into application. But what we are doing with any science, with any discovery will be useful, maybe not in our lifetime, but it will be useful one day to actually make a difference in this world. And this is my dream. It's kind of ideal. I feel that I can actually try to influence the younger people and make them realize this very early on in their life. I hope to do this.
Well, it's good to dream always, right?
Yeah, yeah.
But you'll be fine. You'll stay a scientist, I have no doubt. Julia, have you read any good books lately?
Truth be told, I'm in a book club, and I did not read the last book, which was supposed to be Exit West, and I've heard it's very good. But I'm supposed to be reading Ready Player One, which I've heard is very good. So that's on my nightstand.
Ankur, you've been here the longest of the three, right?
Nope.
So you've been there the longest?
That'd be me.
All right, so then, well, anyway, I'll ask you, what do you do for fun, Ankur?
Oh, what do I do for fun? So I always tell people my job and my number one hobby is doing science. So like, if I can, I'm trying to be at the bench as much as possible. Every day it's getting harder and harder, which just means I'm not doing anything at home, basically. I'm doing a lot of science. And so, yeah, the main hobby is that. But if I get time, I like to go rock climbing as well. So that's the other hobby I try and keep up.
Shindong, what a new student comes to your lab, what's the best advice you can give them?
Yeah, this is a hard question. What's the best advice I can give them? So maybe so this is something I learned at the very beginning of my PhD. Our conclusions are only as strong as our data. So it's very important to have high quality data. But on the other hand, we don't have to do every experiment in the most perfect way. And let me tell you one example. So at some point, so I knew this at the beginning of my PhD, and I tried to do everything perfect. I would find the best controls and do the controls first, and wait until I have everything and do my real experiment. Find the best, find the most elegant way to do a test, and only in the end found that it wasn't so interesting. So my advice to my students is that it's very important to have high quality data. But when you are doing some exploratory research, when you are not sure whether it will work, it's okay to do a pilot experiment to see if it works. And you will have the opportunity to do your perfect experiment if there is something interesting to pursue. Yeah.
All right. Well, we're out of time. So let me just tell you that you can find this podcast and all our others at microbe.tv. You can send questions and comments to TWiM at microbe.tv. If you'd like to support us, which some people do financially, you can go to microbe.tv/contribute, and you can find ways to do that. And I want to thank my guests for today. Shindong Wang, thank you so much.
Thank you.
Ankur Dalia, thank you so much.
Thanks for having me.
Julia Van Kessel, thank you.
Thank you.
Also want to thank this young lady who's standing here, Jolene Ramsey, for getting this all initiated. I met her at ASM in New Orleans earlier this year. She said, would you like to come to Bloomington? And that was the beginning. And she really organized it all and made sure it happened and made a great poster and everything. So thank you very much. And I want to thank the departments here that have participated in my day and so forth. And I want to thank the American Society for Microbiology for their support of TWiM, as well as Ray Ortega, who does a lot of post-production for us. Thanks for listening, everyone. Oh, by the way, I'm Vincent Racaniello. You can find me at virology.ws. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.